Students, let's discuss today about enzymes. Now this enzyme was coined by the scientist. The name is Hun. He is the scientist who first coined this enzyme name. Now enzymes are what? Colloidal organic macromolecules which are mostly proteinaceous in nature. So they are protein in nature. These enzymes are called biocatalysts. These enzymes are called as biocatalysts that speed up or enhance the biochemical reaction. Except these protein in nature, except one enzyme, that is ribozyme, which is not a protein. The ribozyme. So ribozyme is a catalytically active RNA. So it is a ribonucleic acid. Now enzymes are water soluble. Enzymes are water soluble in nature, and enzymes are very active between 10 degree centigrade to 40 degree centigrade, and also enzymes are active at pH. pH is very important for the activation of enzymes. So these are certain characteristics of enzymes. So let's go to now the structure of enzymes. In the structure of enzymes, so properties of enzymes are clear, that enhance the rate of chemical reaction without themselves being changed, and enzymes are very efficient in small amount. Enzymes are highly specific, and each of them, so they're specific in nature also, very specific. That means one enzyme can control only one reaction, and enzymes are denatured by heat, and that is very like human enzymes are active at 35 to 40 degrees centigrade, as our body temperature is 37 degrees centigrade, and denatured at, so enzymes can be denatured if it exceeds 55 degree centigrade, then enzymes cannot function. But the enzymes of certain bacteria can withstand, that is bacteria, that is thermostable enzyme is found in this, and that can tolerate the temperature that is more than 70 degree centigrade also. So it is very important that this quality of enzymes isolated from the organism who normally live under extremely high temperature, that is hot thermal vents under seawater and sulfur spring. These enzymes have the ability to be stable and retain their catalytic power even at high temperature. So let's talk about what are the types of enzymes, and there are 6 categories of enzymes. The first one is oxidoreductases, second one is transferase, third one is lyase, fourth one is ligase, and isomerase, hydrolysis. So these are the categories of enzymes. Now the first one is oxidoreductases, that removes and add electrons, oxidation and reduction reactions. Transferase is a transfer of functional group, carbon group, phosphate group, sulfur containing group. Hydrolysis is the hydrolysis reaction, is breaking down of introduction of water and the breakdown of the covalent bonds. Then lysis catalyzes the cleavage of specific covalent bonds and removal of groups without hydrolysis. Isomerase catalyze the isomerization reactions, catalyze that rearrangement of molecular structure of isomers. And ligase is the formation of bonds with ATP cleavage, that is joining two components, that is done by this ligase. So these are the types of enzymes, category of enzymes. Next comes to the nature of enzyme action. So nature of enzyme action, there is a equation E plus S gives EP, and that again breaks down to E plus P. So let's understand this. Each enzyme E has a substrate binding site in its molecule in order to form a highly reactive enzyme substrate complex. So this is enzyme substrate complex, then changed to enzyme product complex. The hypothesis is called as lock and key mechanism. So in the lock and key theory is very important to describe this nature of enzyme action. It is discovered by Emil Fischer. Lock and key is given by Emil Fischer. That's a scientist. So the catalytic cycle of an enzyme action can be described in the following steps. What are the steps which describe this? First, the substrate binds to the active site of the enzyme to alter its shape, and the binding of the substrate induces the enzyme to alter its shape so that it can fit more tightly around the substrate. And the active site of the enzyme, now in close proximity of the substrate, breaks the chemical bonds of the substrate, due to which the new enzyme product complex is formed. And the last step is the enzyme releases the products of the reaction, and the free enzyme is ready to bind to another molecule of the substrate so that it can run through the catalytic cycle once again. So how it normally happens, if you take an enzyme, if this is the shape of the enzyme. Now this one is the active site where and this is called an allosteric site. So enzyme normally has this type of sites, active sites and allosteric sites. These active sites, the substrate joins. So this is the active site where the substrate joins. This is the substrate, and then this is called enzyme substrate complex. Later these bonds break and the enzyme is again free. The enzyme is set free, and then this substrate is broken down into products. Substrate is broken down into products, and another one like this. So like these two products are formed. So this is enzyme has active site, so this catalytic cycle of enzyme is possible. And remember, after each reaction the enzyme is not used up, it sets free. Now what is the concept of activation energy? If you say what is activation energy, so there is a graph that will tell you what is this activation energy. So here you can see this is happening, and this is without enzyme. Now here what is this progress of reaction? This one x-axis is the progress of reaction, and this side is potential energy. Now activation energy without enzyme is this, and activation energy with enzyme is required this. So see, the product is formed very fast when enzyme is present, but product to be formed takes very much more time without enzyme. So when P is at a lower level than S, the reaction is exothermic or spontaneous. So this one is called transition state, and this product is formed here. Product is P, and here the substrate called S. This side substrate is called S, and this is the activation energy without enzyme. So this is your without enzyme, and this one is with enzyme. With enzyme. Without enzyme, with enzyme. So stability is related to the energy status of the molecule or the structure. It is pictorially presented through a graph given below. This is in the graph y-axis is the potential energy content, and the x-axis represents the progression of the structural transformation. And when P is at lower level, then S reaction is exothermic or spontaneous. So no need of external supply of energy. But when P is at higher level than S, reaction is endothermic and energy requiring reaction. So this is the activation energy count. It proves that whether it is exothermic or endothermic, the S has to go through a much higher energy state, that is transition state. This difference between the average energy content of S from that is transition state is called activation energy. So here you can see the difference in the timing to give the product. So that this difference is the activation energy between these two states. Next, come to the factors affecting enzyme activity. So what are the factors that affects the enzyme activity? The first one is the temperature, then pH, then substrate concentration, then enzyme inhibition. Enzyme inhibition. So let's study the factors affecting enzyme activity. The first one is temperature. Enzymes generally work at a narrow range of temperature and pH. Each enzyme shows the highest activity at a specific temperature and pH called optimum temperature or optimum pH. Like salivary amylase function at 37 degrees centigrade. So here you will get a graph like this. In the both side temperature as well as pH, this type of graph is there. That means what it says, this is the peak point. This is the peak point. This one is a peak point for enzyme as well as pH. So during low temperature, the enzyme is preserved temporarily in an inactive state. So here the low temperature, this is considered as temperature, and this is considered as pH in x-axis. Then you will get the working of enzymes. That is, during low temperature, if the enzyme is preserved temporarily in an active state, whereas at high temperature the activity gets destroyed because proteins denatured at high temperature. Now if you talk about the pH also, change in pH of the medium alters the physical structure of the enzyme molecule for denaturation, decreases enzyme activity. And the peaks in the graph shows maximum and optimum is enzymatic activity. The next one is concentration, that's substrate concentration. Now substrate concentration, there is a graph again showing this type of diagram is there. Now here, see, it is written lower one, the x-axis is substrate, and this is V max by 2. This is the substrate concentration, and this one is the V max. This is called Km. So let's describe this substrate concentration. With the increase in the concentration of the substrate, the velocity of the enzymatic reaction rises at fast, and thus the reaction finally reaches a maximum velocity, that is V max. And V max is the maximum velocity which does not exceed by any further rise in the concentration of the substrate. This is because at this stage the enzyme molecules become fully saturated and no active site is left free to bind an additional substrate molecule. So, and this is the phase where you can say half of that V max, where maximum velocity is observed, and that's called Michaelis constant. Next is enzyme inhibition. Certain products are there which inhibits the enzyme function, and the enzyme activity is also very sensitive to the presence of specific chemicals that shuts off the enzyme when they bind to the enzyme, and that's called what? Inhibition. So when enzyme activity is shutting down and that does not work, that is called inhibition, and the inhibitors are those components which shuts the enzyme activity. Now that is the cyanide, heavy metal, salts, these are coming under inhibitors. And the factors that activate the activity of enzymes are called activators. So opposite of this is activator. So one is inhibitor and one is activator, and activator are like magnesium, calcium, zinc. So all these are activator. Even HCl is also there, hydrochloric acid. Inhibitors are cyanide. Is a common example of inhibitor. Along with that, heavy metal, salts. So these are the inhibitors. Next we will discuss the structure of enzyme. And the structure of enzyme is normally known as, it is a hollow enzyme which contains two parts, an active part, that is a protein part, and is a non-protein part. Now this protein part is called as apoenzyme, and this non-protein part is again of many types, that is, so non-protein part is prosthetic group. So these are apoenzyme is the protein part, and the simple enzyme, that is hollow enzyme, and non-protein parts are called cofactor. And this cofactor can be of again three types. That may be a coenzyme, that may be a prosthetic group, and one more is there, that is metal ions. Metal ions. So these structures. Now what is this apoenzyme? This is protein part, and prosthetic group is the tightly bound to the apoenzyme. If the component is very tightly bound and that is organic in nature, then it is called prosthetic. And coenzymes, this associated with apoenzyme only during the course of catalysis, and they are generally organic in nature, and they are mostly acting as carrier or chemical group in atoms and reactions. So like coenzymes are FAD, NAD, so all these are coenzymes. Whereas prosthetic groups like heme is the prosthetic group, and the part of active site. Heme is to globin, hemoglobin. Heme is the prosthetic group. So this completes the study of enzymes. Go through NCERT. Thank you.